ಶುರು ಮಾಡಿದೆ ಸರ್.
ಮಾಡ್ಬೋದು ಅಂದ್ರೆ ಮಾಡೋಣ ಸರ್.
ಮಾಡೋಣ ಸರ್. ಸ್ಟೂಡೆಂಟ್ಸ್ ಜಾಯ್ನ್ ಆಗ್ತಾ ಇದಾರೆ.
ಆಗ್ಲಿ ಒಂದು 5 ನಿಮಿಷ, ಇನ್ನೂ 3 ನಿಮಿಷ ಇದೆ. ಬರ್ಲಿ ಸರ್, ತೊಂದ್ರೆ ಇಲ್ಲ. ನೀವು ಹೇಳಿ ಯಾವಾಗ ಅಂದ್ರೆ ಅವಾಗ ನಾನು ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ತೀನಿ.
ಅಲ್ಲ, ತಮ್ಮ ಬಗ್ಗೆ ಒಂದು ಇಂಟ್ರೊಡಕ್ಷನ್ ಕೊಡ್ತೀನಿ ಸರ್. ಆ ಟೈಮ್ ಕರೆಕ್ಟ್ ಆಗುತ್ತೆ.
ಓಕೆ.
ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಸೊ ಗುಡ್ ಈವ್ನಿಂಗ್ ಎವ್ರಿಬಡಿ. ಸೊ ಇಂದಿನ ವಿದ್ಯಾರ್ಥಿ ಪ್ರತಿಭಾಶೋಧ ಯೋಜನೆಯ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದ ಉಪನ್ಯಾಸಕರಾದಂಥ ಡಾಕ್ಟರ್ ಎಸ್.ಆರ್. ರಮೇಶ್ ಸರ್ ಅವರನ್ನ ಈ ಒಂದು ಉಪನ್ಯಾಸ ವೇದಿಕೆಗೆ ಸ್ವಾಗತಿಸುತ್ತಾ, ವೆಲ್ಕಮ್ ಟು ಯು ಸರ್.
ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
ಅದೇ ರೀತಿ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಾಗಿರ್ತಕ್ಕಂಥ ನನ್ನ ಎಲ್ಲ ನೆಚ್ಚಿನ ವಿದ್ಯಾರ್ಥಿಗಳು, ಟೀಚರ್ಸ್, ಶಿಕ್ಷಕರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ನಮ್ಮ ನೆಚ್ಚಿನ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ಎ.ಎಂ. ರಮೇಶ್ ಸರ್ ಹಾಗೂ ಅಕಾಡೆಮಿಯ ನಿರ್ದೇಶಕರಾದಂತಹ ಡಾಕ್ಟರ್ ಎ.ಬಿ. ಬಸವರಾಜ್ ಅವರನ್ನು ಕೂಡ ಈ ಒಂದು ವೇದಿಕೆಗೆ ಸ್ವಾಗತಿಸುತ್ತಾ, ನಮ್ಮ ಡಾಕ್ಟರ್ ಎಸ್.ಆರ್. ರಮೇಶ್ ಸರ್ ಬಗ್ಗೆ ಒಂದು ಕಿರು ಪರಿಚಯ ಮಾಡಿಕೊಡಲಿದ್ದೇನೆ. Sir got his MSc and PhD from Mysore University and postdoctoral research Institute for Genetics, Ruhr University, Bochum, Germany, and a DOD fellowship. And also is DOD visiting fellow Institute for Genetics, Ruhr University, Bochum, and Heinrich Ein University, Düsseldorf, Germany. And so working as a research officer, Vector Control Research Center, ICMR, Pondicherry in 1980. And working as a lecturer in the Sericulture University of Mysore in 1981. And working as a lecturer in Zoology, Mysore University in 1982. A senior lecturer in Zoology, University of Mysore in 1987. And reader in Zoology, University of Mysore, 1994. Professor of Zoology, University of Mysore, 1999 to 2016, and now visiting professor, University of Mysore, August 2016 to February 2017. And sir, I have field specialization and Drosophila genetics, and having 35 years teaching experience and 41 years experience in research side. And offered some courses: environmental biology, cytogenic genetics, psychology, cell and molecular biology, genetics, immunology for master degree students of zoology. And sir offers some courses, immunology and genetics for master degree students of genetics, and also for master degree students of microbiology, as well as course in population biology for master degree students of environmental science, University of Mysore. And sir have research projects in principal investigator, co-investigator as on 8, funded by Department of Atomic Energy, UGC, Department of Science and Technology. Sir published 60 national and international journals and international research reports of 33 and conferences abstracts 59. And popular science articles in Kannada 12, and number of PhD successfully guided 13 plus 1 as co-guide. And sir published one book, Immunology, publisher Mr. McGraw Hill Education. And sir as a associate editor, Global Science and Technology Forum in Singapore. Reference for national and international journals in scientific reports like Nature Publishing Group, Journal of Natural Science, Biology and Medicine, Gulwa, Journal for Functional Reports, and Journal of Insect Science. And the curricular activities development member of organizing committee for workshop, symposiums, and refresher courses about 40. And working as a chairman, member of committee, UGC 11, CFTRHO, University of Mysore and other universities 75, and invited lectures 85. Okay, thank you, sir. Over to you.
Thank you very much, Siddhegowda. So nice of you. And I would like to, uh, welcome our youngsters for this a kind of discussion meeting. Don't think that I am giving you a lecture on some particular aspect. Feel free to interact with me at any point of time so that it will be more of a kind of interaction rather than one-way lecture on some particular topic like today, dynamics of cell division anta. Uh, Students can respond over mic, Siddhegowdra.
ಸರ್ ಇಲ್ಲ ಅಕಸ್ಮಾತ್ ದೇ ಹ್ಯಾವ್ ಟು ಟೇಕ್ ಚಾನ್ಸ್ ನಾನು ಇಲ್ಲಿ ನಾನು ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಡ್ತೀನಿ ಸರ್ ಸೊ ಅದರ್ವೈಸ್ ಪುಟ್ ದ ಕ್ವಶನ್ಸ್ ಇನ್ ಚಾಟ್ ಬಾಕ್ಸ್ ಸರ್
ಚಾಟ್ ಬಾಕ್ಸ್
ಎಸ್ ಸರ್
ಮಾಡ್ಬೋದಾ?
ಎಸ್ ಸರ್ ಎಸ್ ಸರ್
ಅದು ಎನೇಬಲ್ ಮಾಡಿಬಿಟ್ರೆ ಅವ್ರಿಗೆ ಬಿಕಾಸ್ ಐಲ್ ಬಿ ಆಸ್ಕಿಂಗ್ ಸಮ್ ಕ್ವಶನ್ಸ್ ಡ್ಯೂರಿಂಗ್ ಮೈ ಲೆಕ್ಚರ್,
ಹಾಂ ಹಾಂ
ಸ್ಮಾಲ್ ವೆರಿ ಜನರಲ್ ಕ್ವಶನ್ಸ್. ಅದಕ್ಕೆ ಅವ್ರು ರೆಸ್ಪಾಂಡ್ ಮಾಡಿದ್ರೆ ಚೆನ್ನಾಗಿರುತ್ತೆ.
ಇಲ್ಲ ನೀವು ಮಾತಾಡಿ ಸರ್
ಅದರ್ವೈಸ್
ಮಾಡದಂತ ಸಂದರ್ಭದಲ್ಲಿ ಇಲ್ಲಿ ನನಗೆ ಇಲ್ಲಿ ಕಳಿಸ್ಕೊಡ್ತಾರೆ ಸರ್ ಇಲ್ಲಿ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಚಾಟ್ ಮಾಡ್ತಾರೆ ಅವರು
ಓಕೆ ಓಕೆ. So let me start with today's topic. Actually, ಡೈನಮಿಕ್ಸ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್ ಅಂತ ಕನ್ನಡದಲ್ಲೂ ಮಾತಾಡ್ಬೋದಲ್ವಾ?
ಸರ್ ಹೌದು ಕನ್ನಡ ಇಂಗ್ಲಿಷ್ ಬೋತ್ ಸರ್ ಪ್ಲೀಸ್.
ಓಕೆ ಫೈನ್. ಡೈನಮಿಕ್ಸ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್ ಅಂತ ಬೇಸಿಕಲಿ ಇಟ್ ಈಸ್ ಮೆಕ್ಯಾನಿಸಮ್ ಹೌ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಸೆಲ್ ಡಿವೈಡ್ಸ್ ಅಂತ ಅಂದ್ಬಿಟ್ಟು ನಿಮ್ಗೆಲ್ಲ ಅರಿವು ಇರಲಿ ಅಂತ ಅಂದ್ಬಿಟ್ಟು, ಬಿಕಾಸ್ ದಟ್ ಈಸ್ ದ ಬೇಸಿಕ್ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಆಫ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ಎ ಝೈಗೋಟ್ ಆಫ್ಟರ್ ಫರ್ಟಿಲೈಸೇಷನ್ ನಿಮ್ಗೆಲ್ಲ ಗೊತ್ತಿದ್ದಂಗೆ. ಸೊ ಫ್ರಮ್ ಸಿಂಗಲ್ ಸೆಲ್ಡ್ zygote, multicellular organisms arise, like for example human beings. So this is possible by two different means. ಒಂದೇನು ಅಂತಂದ್ರೆ by means of cell division there will be growth, and then that is followed by you know growth of the divided cell. ಆ ಥರ ಎರಡು process ಅಲ್ಲಿ we become from baby we grow as adult and then reach old age and so on and so forth. ಸೊ ಇದು ಆಗ್ಬೇಕಾದ್ರೆ ನಮಗೆ there should be continuous activity of the cells within the body without which life is not possible. So in this context, dynamics of cell division ಅಂತ ಅಂದ್ಬಿಟ್ಟು ನಾನು ಫಸ್ಟ್ ಚೂಸ್ ಮಾಡಿಕೊಂಡು ಒಂದ್ ಸ್ವಲ್ಪ ನಿಮ್ಗೆ ಮಾಹಿತಿನ ಹಂಚ್ಕೋಬೇಕು ಅಂತ ಅಂದ್ಬಿಟ್ಟು sharing my knowledge with you youngsters. And I do not know how far or what is the depth of my dealing with you as far as cell division is concerned. I am still is a big question mark for me, because I've been teaching this dynamics of cell division for master degree students of zoology or biotechnology or microbiology or biochemistry for more than 35 years. So actually, because of the time available, everything I had to condense in 1 hour. ಸಿದ್ದೇಗೌಡರು ನನಗೆ 1 hour ಮಾತಾಡಿ ಸಾಕು ಅಂತಂದ್ರು. ಸೊ ಹಾಗಾಗಿ all that is condensed, and what is important for you at your level, I'm only giving you information, and later on some excitements about cell division and how important it is for your future career, I would like to highlight. So starting from the basics, you can know. So I would like to project my slides. Say cell division as such, basically you can think of whenever you talk about cell division, basically you will see in your textbooks two types of cell division. One is mitosis, and the name that is given to this particular cell division is thread-like appearance of the chromosomes, mitosis antivi. Sorry, this is making a lot of noise. I should click off that noise anyway. ಪ್ಲೀಸ್ ಬೇರ್ ವಿತ್ ಮೀ. ಇನ್ನೊಂದು ಮಿಯಾಸಿಸ್ ಅಂತ ನಿಮ್ಗೆಲ್ಲ ಗೊತ್ತಿರುತ್ತೆ. ಮಿಯಾಸಿಸ್ ಬೇಸಿಕಲಿ ಈಸ್ ಎ ಗ್ರೀಕ್ ಟರ್ಮ್ ದಟ್ ಸಿಗ್ನಿಫೈಸ್ ಡಿಮಿನ್ಯೂಷನ್. ಮೈಟಾಸಿಸ್ಗೆ ಮಿಯಾಸಿಸ್ಗೆ ಡಿಫರೆನ್ಸ್ ಇರೋದು, ಒಂದು ಮೈಟಾಸಿಸ್ ಅಲ್ಲಿ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ಸ್ 10 ಇದ್ರೆ 10 ಇರುತ್ತೆ. ವೆನ್ ದಿ ಸೆಲ್ ಡಿವೈಡ್ಸ್ ಇಂಟು 2 ಫ್ರಮ್ 1 ಅಂಡ್ ದಿ ಡಿವೈಡೆಡ್ ಸೆಲ್ ಫರ್ದರ್ ಗೋಸ್ ಆನ್. ಒಂದು ಸೆಲ್ ಇರೋದು 2 ಆಗುತ್ತೆ, 2 ಇರೋದು 4 ಆಗುತ್ತೆ, 4 ಇರೋದು 8 ಆಗುತ್ತೆ. ದಟ್ಸ್ ಹೌ there is a geometric increase in the number of cells during the process of mitotic division. Meiosis ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, the chromosomes that are present in a cell is divided into half. Or there will be reduction in the number of chromosomes, and that's why it is called as a diminution, ಅಂದ್ರೆ reduction in the number of chromosomes. It's also called as reduction division. Meiosis ಕೊನೆಗೆ reduction division ಅಂತ ಹೇಳ್ತೀವಿ. Um, uh, to tell you in a textbook language, ಮೈಟಾಸಿಸ್ ಅಲ್ಲಿ 2n ಏನಿರುತ್ತೋ, ಇದು ಡಾಟರ್ ಸೆಲ್ಸ್ ನಲ್ಲಿ 2 ಸೆಲ್ಲು ಒಂದು ಮದರ್ ಸೆಲ್ ಇಂದ ಡಿವೈಡ್ ಆದಾಗ ಡಾಟರ್ ಸೆಲ್ಸ್, 2 ಡಾಟರ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದಿ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ ರಿಮೇನ್ಸ್ ಸೇಮ್. ಸೊ 2n ನಂಬರ್ is retained, while in meiosis 2n becomes n number. That is actually mitosis is seen in all somatic cells, whereas meiosis is confined to germ cells. Wherever there is production of gametes, the chromosome number is reduced, and as a consequence of fertilization, once again, the chromosome number is restored in the zygote. Okay, so the highlights of mitotic division are given here. Two daughter cells are produced which are genetically equivalent to a mother cell. So mitosis, the daughter cells that are obtained, they will not differ from one another, unlike in case of meiosis. ಅದೇನು ಅಂತ ಹೇಳ್ತೀನಿ. So parental chromosome number is maintained in mitosis, that is 2n remains 2n. If the mother cell has got 2n is equal to 10, the daughter cell also will have 2n is equal to 10. And mitosis rarely goes wrong. So it's a perfect kind of division. Unless there is something which is coming in the way of functioning of dividing cell, it rarely goes wrong. Failure of mitosis produces unviable cells, except in case of cancer. ಅಲ್ಲಿ ಏನೇನೋ ಆಗೋಗ್ಬಿಡುತ್ತೆ, that you need not worry. And mitosis stops. If mitosis stops, the organism quickly grows old, wear away, and becomes weak, and as a consequence, the organism will die. This is true not only in case of humans, but also in case of animals or plants or wherever you see cell division. Meiosis ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, you'll see that the consequence of meiotic cell division is genetic continuity is maintained, but at the same time, there is introduction of variation in the genetic content. The reasons are many how variation occurs. I have listed 1, 2, 3, and need not worry. And one of the very noted feature is of meiosis is there will be reduction in the parental chromosome number. As I told you, if 2n is equal to 10, the resulting daughter cells out of meiosis, they will have 2n, n number, that will be chromosome number will be reduced to 5. And there will be variation in predictable frequency as a consequence of meiosis. Meiosis ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಅಲೀಲ್ಸ್ ಸೆಗ್ರಿಗೇಟ್ ಆಗುತ್ತೆ. ಆಮೇಲೆ there will be union of gametes of dissimilar genotypes. So genetic variations ಇದು as a consequence, ಎರಡು gametes unite ಆದಾಗ they will not have similar genotype. So the question is, why mitosis? Why cell division has to occur? Cell division has to occur because it is the principal source of growth. As I told you, from the childhood, when we grow up into adults, obviously mitosis should occur, which increases the size of the organism and also the size of the organs within, etc., etc. That is only possible with the consequence of mitotic division. And mitosis is the only mechanism for replacement of dead and gone cells. ಈ ಸೆಲ್ಸ್ ಏನು ಉತ್ಪತ್ತಿ ಆಗುತ್ತೆ ಎಲ್ಲಾದರೂ ಆಗಲಿ, ನಾನು ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ ಆಮೇಲೆ. Many of the cells will undergo death because its life is limited. Dead and gone cells have to be replaced with similar kind of cells, and that mechanism is only possible with the occurrence of mitosis, mitotic cell division. So cell division ಆಗುವಾಗ many forces, many molecules are involved. We'll take one cell into consideration, and I'll show you how different molecules are involved, or how different kinds of mechanisms are involved in making one cell into two cells. One example I can give you why mitosis andre, average young healthy human adult ಅಲ್ಲಿ per second you can see that there are about 20 million mitotic divisions that are occurring per second. So even if you are simply sitting and listening to my lecture, you unknowingly something is happening in your body that is in relation to mitotic cell division. It is estimated that 20 million mitotic divisions occur per second in an average young healthy human adult. So you can imagine the power of mitosis or the requirement of mitotic cell division, because the organism has to live, organism has to survive. Okay, so where and all these mitotic divisions are occurring in your body is listed here. You can see 20 million mitotic divisions together. They occur in the bone marrow, they occur in the skin, in the lining of intestine, lymph nodes, spleen, hair follicles, and in the vicinity of the wounds. Not only this, several other places. I have listed only a couple of them. So that kind of activity is required to maintain the tissues, to maintain the body, to maintain the dynamics of the living organism. One example ಕೊಡ್ತೀನಿ. If you take your blood basically, as you know, it contains red blood cells and white blood cells in definite quantities. So as far as RBC is concerned, red blood cells are concerned, you can see that women have got 4.2 to 5.4 millions of red blood cells per microliter of blood, per microliter of the blood. Men have got a little more higher, 4.7 to 6.1 million RBCs per microliter, and children also will have a different number, 4.1 to 5.5. And this RBC, the average lifespan of the RBC, that is, when the RBC is produced, it will not live for lifetime. After 150 days, 115 days to 120 days, the red blood cell dies. It will die. Okay. Similarly, you can see the white blood cells, which are actually important as far as immunity is concerned, or as far as your disease resistance is concerned. The number varies from 4,000 to 11,000 cells per microliter of blood in all males, females, and children, etc., etc. And surprisingly, the lifespan of these white blood cells is much lesser as compared to red blood cells. The lifespan of white blood cells or white blood corpuscles varies from 13 to 20 days. Roughly, you can take it as 3 weeks, and after 3 weeks, these cells will die. Okay. And whenever you have gone to a pathologist or a doctor for some reason, he would say, get your TC done. TC ಅಂತಂದ್ರೆ total count of your blood cells, RBC, WBC, and differential count ಅಂತಂದ್ರೆ ಯಾವ ಥರದ್ದು. As far as WBC is concerned, There are 5 different types of white blood corpuscles: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Now, all these WBC, once they are produced, they will not live for lifetime, but they will die frequently. Once in 3 weeks, any new cell will die. So that being the case, these numbers, total numbers, have to be maintained within the body for life processes to occur. Without blood cells, nothing can work and nothing is possible. Especially, if there are no RBCs, will not be able to breathe because there will be, oxygen is supplied by RBC to various tissues. And if RBC's count comes down, then there won't be any supply of oxygen to various tissues. And as a consequence, you know very well, will not be able to survive. That is what happened during COVID time. Absence of oxygen, many people died. No, oxygen is very important, for life processes to occur. So that being the case, due to this limited lifespan of RBC and WBC, these cells have to be constantly replenished in such a way that these numbers have to be maintained. See, when there is reduction of RBC number in your body, that leads to anemia and then so many other complications. If there is reduction in the white blood corpuscles in your circulating blood, then also it leads to different kinds of complications, such as the resistance to disease is lost. Not only that, various other functions that are associated with different kinds of WBCs also will hamper the healthy living of anybody who are unable to maintain this total count of WBC. So, that being the case, you can know that these blood cells that have got limited life have to be replaced by similar kind of cells, and that number has to be maintained all the time. Even beyond that, ಈಗ WBC count ಹೆಚ್ಚಿಗೆ ಆಗೋಗ್ಬಿಟ್ರೆ, that also is a pathological condition, especially in case of blood cancer. The number shoots up very high, and then that will lead to different kinds of problems. So it's a fine balancing system of maintaining these different kinds of cells in specified number or the normal range, which actually enables you to have a healthy life. ಸೊ ಇದನ್ನ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತಾ ಇದ್ದೀನಿ, ವೈ ಮೈಟಾಸಿಸ್ ಅಂತ ಅಂದ್ಬಿಟ್ಟು. when the dead cells have to be replaced, the new cells have to be produced, and that production of new cells is possible as a consequence of mitotic division of there is a source for that within the bone marrow. I'll show you that figure. So you can see that ಇಲ್ಲಿ ಎಲ್ಲಿ ತೋರಿಸ್ಬೇಕು ಅಂತಂದ್ರೆ, ನಿಮಗೆ ಇಲ್ಲೊಂದು ಬ್ಲೂ ಕಲರ್ ಸೆಲ್ ಇದೆ. ಇಟ್ ಈಸ್ ಆರ್ಟಿಫಿಷಿಯಲಿ ಕಲರ್ಡ್. ಸೆಲ್ಫ್ ರಿನ್ಯೂವಲ್ ಅಂತ ಹೇಳ್ತೀವಿ. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಸ್ಟೆಮ್ ಸೆಲ್. The stem cells are present in the bone marrow. So as a consequence, it so happens that these stem cells divide. They undergo constant division, and uh, they divide to produce once again similar kind of cell, stem cell, or the divided cell will get into one of these two lineages. So on the top, you can find one blue cell, and then the bottom another blue cell. So that is called as um, production of two lineages of blood cells. One lineage is called a lymphoid lineage. The other one is called myeloid lineage. ಅದೆಲ್ಲ ನೀವು ಮರ್ತ್ಕೊಂಡ್ಬಿಡಿ. ಇದು ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ದೀಸ್ ಡಿಫರೆಂಟ್ ಕೈಂಡ್ಸ್ ಆಫ್ ಸೆಲ್ಸ್ ಆರ್ stem cell dividing into to give rise to stem cell. See, stem cells have to be present all the time in the bone marrow. ಅದು ಒಂದು ಡಿವೈಡ್ ಆಗಿ ಇನ್ನೊಂದು ಸ್ಟೆಮ್ ಸೆಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. That's how it also keeps dividing and dividing continuously, so that the divided products of the stem cells will either get into lymphoid lineage or myeloid lineage of blood cells. These are precursor cells from which there is differentiation of various kinds of blood cells. So it may be lymphocytes, or it may be monocyte, or it may be neutrophil, eosinophil, basophil, etc. So different kinds of blood cells. What you see in the blood ultimately is a consequence of continuous division and differentiation of the divided cells from stem cells, and they get into various kinds of lymphoid tissues, and also they also get into the blood, and in the blood and in the lymphoid tissues they will perform their functions. ಸೊ ಇದೇನು ಅಂತಂತಂದ್ರೆ ಈ ನಂಬರ್ನ ಮೇಂಟೈನ್ ಮಾಡಬೇಕು ಆಲ್ ದಿ ಟೈಮ್. ಸೊ ದಿ ಹಿಯರ್ this is the source of replacement of dead and gone cells. That is, stem cells divide to give rise to stem cells, which undergo differentiation to finally result in different kinds of blood cells. We call them as effector cells, and that includes RBC also. See, ಕೆಳಗಡೆ ನೀವು ನೋಡಿದ್ರೆ ಎರಿಥ್ರೋಸೈಟ್ ಅಂತ ಇಲ್ಲಿದೆ. So erythrocyte originates from erythroid progenitor, which belongs to myeloid stem cell lineage. Okay. So ನಮ್ಮ ಬೋನ್ ಮ್ಯಾರೋದಲ್ಲಿರೋದು stem cell will be continuously dividing and differentiating. So all this is possible because of mitotic cell division. This is a somatic cell, and that's how mitotic cell division. ಈಗ ನಾವು ಸೆಲ್ ಡಿವಿಷನ್ ಡೈನಮಿಕ್ಸ್ ಹೇಳ್ಬೇಕಾದ್ರೆ ಮೋಸ್ಟ್ ಆಫ್ ದಿ ರಿಸರ್ಚ್ ಇಸ್ ಡನ್ ಆನ್ ದಿ ಸೊಮ್ಯಾಟಿಕ್ ಸೆಲ್ಸ್. ಅದಕ್ಕೆ ಮೈಟಾಟಿಕ್ ಡಿವಿಷನ್ ಅಂತಾನೆ ಹೇಳ್ತೀವಿ. ಮಿಯಾಸಿಸ್ ಇಸ್ ಅನದರ್ ಪ್ರಾಸೆಸ್. ಅದರಲ್ಲೂ ಬೇಸಿಕಲಿ ವೆನ್ ಇಟ್ ಕಮ್ಸ್ ಟು ಕ್ರೋಮೋಸೋಮ್ ಮೂವ್ಮೆಂಟ್, ಕ್ರೋಮೋಸೋಮ್ ಸೆಗ್ರಿಗೇಷನ್, ನಾವು ಅದನ್ನೇ ನೋಡ್ತೀವಿ. But basically, whenever I refer to cell division, it means that it applies to mitotic cell division. Okay. So ಇದು ಒಂದು ಎಕ್ಸಾಂಪಲ್ ನಿಮಗೆ ಕೊಟ್ಟಿದ್ದು. ಇದೇ ತರ in our body, various kinds of cells in various kinds of tissues and organs have to be replaced as a consequence of their death, and replacement has to occur. ಒಂದು ನಿಮಗೆ ಇನ್ನೊಂದು ಇನ್ಸ್ಟೆನ್ಸ್ ಕೊಡ್ತೀನಿ. ಈ ಬೋನ್ ಮ್ಯಾರೋ ಇರೇಡಿಯೇಷನ್ ಅಂತ ಮಾಡ್ತಾರೆ. See, when a mouse or a rat is exposed to radiation, high energy radiations like gamma radiation or X-rays radiation, it so happens that these stem cells in the bone marrow are depleted. Depleted andre they will affect the stem cells in the bone marrow. Suppose you provide longer high energy radiations like gamma radiation, ಅದೇನಾಗುತ್ತೆ, ಅದು ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ ಎಲ್ಲ ಸತ್ತೋಗ್ಬಿಟ್ಟು the source of replacement of the blood cells will be lost. Okay, these are primordial cells. ಇದು ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ ಎಲ್ಲ ಡಿಪ್ಲೀಟ್ ಆಗೋಗ್ಬಿಟ್ರೆ hardly after high energy radiation exposure of the rat or a mouse, say for 1 day or half a day, ಏನಾಗುತ್ತೆ, the source will be lost, stem cell deplete ಆಗುತ್ತೆ. As a consequence, blood cells ಎಲ್ಲ ಮೇಂಟೈನ್ ಆಗಬೇಕಾಗಿರೋ different kinds of cells will not be replaced, or dead and gone cells will be going, and within within 3 weeks the organism will die. So if there is no replacement of the dead and gone cells, so the organism will die, or the rat or the mouse to which radiation is given, that will die without the stem cells. So ಅದನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂತಂದ್ರೆ, ಅದನ್ನ ತೋರ್ಸಕ್ಕೆ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಆರ್ ಡನ್ ಇನ್ ಸಚ್ ಅ ವೇ ದಟ್ ಈ ತರ ರೇಡಿಯೇಷನ್ ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ ಆರ್ಗ್ಯಾನಿಸಮ್ಗೆ. ಸೊ ಫಾರ್ ಡಿಫ್ರೆಂಟ್ ಪೀರಿಯಡ್ಸ್ ಆಫ್ ಲೆಂತ್ ದಿ ರೇಡಿಯೇಷನ್ ಈಸ್ ಗಿವನ್, ಅಂಡ್ ಇಟ್ ಈಸ್ ಮೇಂಟೈನ್ಡ್ ಫಾರ್ so many days without any kind of readjustment. ಅಂದ್ರೆ ರೇಡಿಯೇಷನ್ ಎಕ್ಸ್ಪೋಜರ್ ಆದ್ಮೇಲೆ one week ಆದ್ಮೇಲೆ, if they want to see that the organism should remain alive, ಆವಾಗ ಸ್ಟೆಮ್ ಸೆಲ್ ಇನ್ಫ್ಯೂಷನ್ ಅಂತ ಮಾಡ್ತಾರೆ. These are experimental procedures that are done, based on which only you will be able to make some observations and also predictions and things like that. Small experiments, highly planned experiments can be done. So 1 week ಆದ್ಮೇಲೆ ಸ್ಟೆಮ್ ಸೆಲ್ ಇಂಜೆಕ್ಷನ್ ಕೊಟ್ರೆ, 2 weeks ಆದ್ಮೇಲೆ ಕೊಟ್ರೆ ಸ್ಟೆಮ್ ಸೆಲ್ infusement ಕೊಟ್ರೆ, directly it will be sent to bone marrow, and the bone marrow will be able to take up the task of mitotic division followed by differentiation, and the number is restored, the organism will survive. So ಕೊಡದೇನೆ ಹೋಗ್ಬಿಟ್ರೆ ಏನಾಗುತ್ತೆ, 3 weeks there is no source for uh, you know replacement of dead and gone cells. As a consequence, the organism will die. So all these observations, small experiments ನೀವು ಡಿಸೈನ್ ಮಾಡ್ಬಿಟ್ಟು ವಾಟ್ ಯು ಎಕ್ಸ್ಪೆಕ್ಟ್ ಆನ್ ವಾಟ್ ಬೇಸಿಸ್ ಅಂತ ನಿಮ್ಮ ಮನಸ್ಸಲ್ಲಿ ಇರಬೇಕು. ಸೊ ಹಾಗಾಗಿ ಅದು ಅಟ್ ದಿ ಎಂಡ್ ಆಫ್ ದಿ ಎಕ್ಸ್ಪೆರಿಮೆಂಟಲ್ ಪೀರಿಯಡ್ ಏನಾಗುತ್ತೆ ಅದಂತ ಒಂದು ಡೇಟಾ ತಗೊಂಡು ಯು ವಿಲ್ ಟ್ರೈ ಟು ಕಂಪೇರ್ ವಾಟ್ ವಾಸ್ ದೇರ್ ಅರ್ಲಿಯರ್ ಅಂಡ್ ವಾಟ್ ಈಸ್ ದೇರ್ ಲೇಟರ್, ಅಂಡ್ ದಟ್ಸ್ ಹೌ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಪ್ರೊವೈಡ್ ಆನ್ಸರ್ಸ್ ಫಾರ್ ಮೆನಿ ಕ್ವಶನ್ಸ್ ಇನ್ ಬಯಾಲಜಿ. ಓಕೆ, ಸೊ ಇದೊಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟೆ ನಾನು. ಸೊ ಮೈಟಾಸಿಸ್ ಅಂತಂದ್ರೆ ನಿಮ್ಗೆಲ್ಲ ಗೊತ್ತಿರೋ ಹಂಗೆ ಒನ್ ಸೆಲ್ ಇಸ್ ಡಿವೈಡಿಂಗ್ ಇಟ್ ಟು ಸೆಲ್ಸ್, ಅಂಡ್ ಟಿಪಿಕಲಿ the mitotic process ಅಂದ್ರೆ ಪ್ರೊಫೇಸ್, ಮೆಟಫೇಸ್, ಅನಾಫೇಸ್ ಅಂಡ್ ಟೀಲೋಫೇಸ್ ಅಂತ ನಿಮ್ಗೆಲ್ಲ ಚೆನ್ನಾಗಿ ಗೊತ್ತು. ಸೊ typically the mitotic cycle is completed within 30 to 60 minutes. Once the cell gets into division cycle, division stage, prophase, metaphase, anaphase and telophase timeಗೆ 30 to 60 minutes, half an hour to 1 hour, you'll get 2 cells from 1 cell. In between ಏನಾಗುತ್ತೆ, after the cell divides, the cell will get into interphase. Interphase ಅಂದ್ರೆ resting phase. Interphase ಎಷ್ಟು ದಿವಸ ಇರುತ್ತೆ, ಹೇಗಿರುತ್ತೆ? It varies depending upon the kind of tissue or the organ from where the cells are obtained. It varies from about 6 hours to very long times. Some cells may rest for forever. Andre, they will be active, they will be living. For example, neurons, nerve cells, they are terminally differentiated cells. Terminally differentiated cells, andre, it would have come from the primordial cell, undergone differentiation. Once it differentiates into neurons, neurons will never come back to division cycle again. So neurons is the terminally differentiated cell which dies after some time. After some time andre it may take years. Okay, ಅದು ಇಂಟರ್ಫೇಸ್ ಸ್ಟೇಜ್ನಲ್ಲೇ ಇರುತ್ತೆ. ಯಾವಾಗಲೂ ಒಂದ್ಸಲ ಡಿವೈಡ್ ಆಗಿ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗೋಗ್ಬಿಟ್ರೆ, nerve cells for example, adana, those cells will never undergo division. Okay, so but they will be able to carry out their functions as efficiently as expected. Nerve cells, okay. So possibly most of you have seen these cell stages, dividing cell stages under microscope in our colleges. So where you can see, looking into the kind of nucleus, you'll be able to decide whether it is interphase or prophase, late prophase, metaphase, early anaphase, telophase, and late telophase, etc. But I am sure that most of you would have seen these stages under microscope in your colleges. Either when you make these preparations by the way of ಆನಿಯನ್ ರೂಟ್ ಟಿಪ್ಸ್ ನ ಸ್ಕ್ವಾಶ್ ಮಾಡಿದ್ರೆ, ನೀವು ಕೆಲವು ಕಾಲೇಜಸ್ ಅಲ್ಲಿ ಮಾಡಿಸ್ತಾರೆ ಇದು. ಆನಿಯನ್ ರೂಟ್ ಟಿಪ್ಸ್, ದಿ ಟಿಪ್ ಆಫ್ ದಿ ಆನಿಯನ್ ರೂಟ್ ಗ್ರೋಯಿಂಗ್ ಇನ್ ಟು ಇದು ಐದರ್ ಸ್ಯಾಂಡ್ ಆರ್ ಇನ್ ವಾಟರ್. If you cut that tip and then make a squash and observe under microscope, you are bound to get these stages because that tip is very active in mitotic division. That's how the roots grow. Okay, so ಈ stages ನ ನೀವು ನೋಡಿದಿರ ಅಂತ I presume that you might have seen. So as a consequence, I am bringing you that picture followed by what is happening at the molecular level. So now mitosis ಯಾಕೆ ಇಷ್ಟು ಇಂಪಾರ್ಟೆಂಟ್ ಅಂತಂದ್ರೆ, see, mitosis though is being studied over 100 years from now, more than 100 years ಆಯ್ತು mitosis ಮೇಲೆ ರಿಸರ್ಚ್ ಮಾಡಿ, ಅದನ್ನ ಸ್ಟಡಿ ಮಾಡಿ, ಅದನ್ನ the process before it is understood. We are studying mitosis in different kinds of tissues, different organisms, and under different conditions of existence, etc., etc., for more than 100 years from now. Okay, but it is still a fascinating story of production of two daughter cells from one mother cell that are genetically identical. Okay, so that is the excitement about mitosis or cell division as such. And of course, why we have to study mitosis is that's a typical cell that is dividing. And if we understand every kind of mechanism that regulates or that controls mitotic cell division, possibly, although we have many answers for, you know, a cell going out of control continues to divide and giving rise to cancerous condition. So ಅದು ಯಾಕಾಗತ್ತೆ ಹೇಗೆ ಅದು ಇಟ್ ಬಿಕಮ್ಸ್ ಮ್ಯಾಡ್ ಇನ್ ಡಿವೈಡಿಂಗ್ ಫ್ರಮ್ 1 to 2, 2 to 4, ಎಟ್ಸೆಟ್ರಾ. ಅದಕ್ಕೆ ಆನ್ಸರ್ ಸಿಕ್ಕಿದ್ರೆ ದೆನ್ ವಿ ವಿಲ್ ಕಾಂಕರ್ ದಿ ಕ್ಯಾನ್ಸರ್ ಆಸ್ ಸಚ್. ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಿ ಫಿಸಿಯಾಲಜಿ, the molecular biology, and various other forces that control mitotic cell division. Only then, when we have complete answer, although we know very much about mitotic division and the molecules that are involved, but still we are hopeful to get a control over cancerous cells which go out of control as far as division is concerned. So, ಇದು ಮೈಟಾಸಿಸ್ ಅಂದ್ರೆ ಸೆಲ್ ಡಿವಿಷನ್ ಸ್ಟಡಿ ಮಾಡಕ್ಕೆ this will be an impetus. I'll skip couple of these slides because it is not required. So only to bring you here, you can see that mitosis or cell division is a part of entire process. What we call it as cell cycle. Cell cycle nalli 4 stage irutte. ಅದನ್ನ ವಿಲ್ ಐಡೆಂಟಿಫೈ ಇಟ್ ಆಸ್ G1s, G2 ಅಂಡ್ ಎಂ ಫೇಸ್ ಅಂತ ಅಂದ್ಬಿಟ್ಟು. ಇಂಟರ್ಫೇಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, G1s, G2 ಇರುತ್ತೆ. G1 ಅಂದ್ರೆ ಗ್ರೋತ್ ಅಂಡ್ ಮೆಟಬಾಲಿಕ್ ರೋಲ್ಸ್ ಆರ್ ಕ್ಯಾರಿಡ್ ಔಟ್. ಎಸ್ ಈಸ್ ದ ರೆಪ್ಲಿಕೇಶನ್ ಆಫ್ DNA, the stage at which the DNA is replicated. G2 is the growth and more preparation for a cell to get into mitosis or division cycle. So whatever we study under cell division is only a small part of what is known as cell cycle. Okay, G1 as G2. If you observe this figure, you'll see mitosis. G1 S is the synthetic phase, followed by second growth phase, that is G2. And many times it so happens after mitosis, the cells will get into G0. G0 is that resting stage. ನಾನು ಹೇಳಿದ್ನಲ್ಲ ಇದು ಯಾವುದು neurons, for example, they will exit from the cell cycle. They will exit from the cell cycle and they remain there until they die. Okay. So whatever we are dealing with, prophase, metaphase, anaphase, telophase, followed by cytokinesis, is only a small part of the cell cycle. I'll show you the other one, figure where you can realize the importance of the division cycle or cell division, which is only a part of G1 as G2 phase, G1 as G2 and M phase. Okay. So this is cell cycle. The whole circle is the cell cycle. And you can see only a small region of the cell cycle is occupied by cell division. ಸೊ ನಾವು ಸ್ವಲ್ಪ ಡೀಟೇಲ್ ಆಗೋದ್ರೆ ನಿಮಗೆ ಎಷ್ಟು ಡೀಟೇಲ್ ಬೇಕೋ ಗೊತ್ತಿಲ್ಲ ನನಗೆ. ಪ್ರೊಫೇಸ್ ಇಟ್ಸ್ ಎ ಟ್ರಾನ್ಸಿಷನ್ ಫ್ರಮ್ G2 to M phase, ಅಂದ್ರೆ ಗ್ಯಾಪ್ 2 to ಮೈಟಾಟಿಕ್ ಫೇಸ್. ಎಂ ಫೇಸ್ ಅಂದ್ರೆ ಡೂಪ್ಲಿಕೇಟಿಂಗ್ ಫೇಸ್. ಅದರಲ್ಲಿ ಸ್ವಲ್ಪ ಚೇಂಜಸ್ ವಿ ಕ್ಯಾನ್ ನೋಟಿಸ್ ವಿತ್ ಇನ್ ದಿ ಸೆಲ್. ದಟ್ ಈಸ್ ಸೆಂಟ್ರಿಯೋಲ್ಸ್ ಅಂತ ಇರುತ್ತೆ. ಸೆಂಟ್ರಿಯೋಲ್ಸ್ ವಿಲ್ ಬಿ ಪ್ರೆಸೆಂಟ್ ಆನ್ ದಿ in the cytoplasm. The centrioles will duplicate, and after duplication, ಎರಡಿರುತ್ತೆ actually, 2 pieces that will become 4, and 2 each. They exist in pairs. although they start moving apart and occupy two different poles of the dividing cell. Okay. Asters, they are the radiating structures that come out from the centrioles, and they will be like a sun. Okay. Asters, one kind of tubules. I'll give you the chemical structure of those tubules. And then there are some proteins that actually play a major role in the cell division. Condensin ಅಂತ ಒಂದು protein. So when condensin enters the nucleus in the prophase, chromosome condensation begins. So condensin protein is responsible for condensation of the chromosomes from chromatin to chromosomes. Prophase ಅಲ್ಲಿ ನೀವು ನೋಡಿದ್ರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ chromosomes ಕಾಣೋದಿಲ್ಲ. It's just a chromatin. It's just a chromatin. And they are transformed into chromosomes as a consequence of the entry of condensin into the nucleus. So condensin, when it enters the nucleus, chromosome condensation begins. And of course, the rest of the cell organelles like Golgi or endoplasmic reticulum, antadella, it is fragmented and that disappears. Okay. ಆಮೇಲೆ reformation ಆಗುತ್ತೆ. So these are centrioles. You can see the duplication of centrioles. They exist in pairs where they are tubular structures. So after they are duplicated, they start moving apart. And after movement, it occupies two opposite poles in a dividing cell. So here is a picture to show the function of condensin 1 and condensin 2. Adar jotege, there will be other kinds of proteins like cohesin or topoisomerase that is actually important for various coiling or condensation of chromatin into chromosomes. If you see the left side of this picture, this is how the interphase uh, chromosome will be there. It is not actually called chromosome, but it is called as chromatin, which is a network of DNA along with proteins, namely histones. Yeah. So it starts condensing in presence of condensin, and further condensation is possible in presence of condensin 2, giving rise to late prophase chromosomes, which is still uh, uh, little fuzzy and also loosely organized. And topoisomerase, the presence of DNA topoisomerase enables these late prophase chromosomes to acquire the typical shape of chromosome, what we see in case of metaphase, what we see in metaphase stage. So let me not go into the detail because prophase chromosome in presence of condensin and becomes metaphase chromosome with a process known as supercoiling. And this supercoiling of prophase chromosome or chromatin into metaphase chromosome requires energy, and that energy is provided by adenosine triphosphate. Okay. So ಇದು ಹೇಗೆ ಗೊತ್ತಾಯ್ತು ನಮಗೆ condensin is very essential for condensation of chromatin into chromosome ಅಂತಂದ್ರೆ by experimental procedures you can deplete the condensin in a dividing cell. Okay, prophase ಅಲ್ಲಿ if you if you set up an experiment where you can deplete or take away this particular condensin or prevent the functioning of the condensin protein, it so happens either you inactivate the condensin or take out the condensin. That is possible by various experimental means. Depletion of condensin results in chromosome condensation, which is defective. So ಸರಿಯಾಗಿ ನಮಗೆ ಮೆಟಾಫೇಸ್ ಕ್ರೋಮೋಸೋಮ್ ಅನಾಫೇಸ್ ಕ್ರೋಮೋಸೋಮ್ ಕಾಣೋದಿಲ್ಲ ಅವಾಗ. So that's how you can make inferences that this particular protein is necessary for condensation of the chromosome, etc., etc. ಇದೇ ತರ ನಮಗೆ whatever the information that we have acquired with reference to cell division or dynamics of cell division is based on series of experiments and technical advancements and technical gadgets have helped us to decipher all this kind of information based on the data that is generated. So now we know what is the reason, what are the forces, what are the molecules, what are the mechanisms. ಅದೆಲ್ಲ ಒಂದೊಂದೇ unravel ಮಾಡ್ತಾ ಹೋಗುತ್ತೆ. This is how research will progress in a particular area, finding out different kinds of situations, different kinds of environmental milieu that is provided to a living cell, and different kinds of molecules or ions that are responsible for this one particular event, like cell division. Okay, so this is I gave you I gave it an example for you to appreciate the role of different molecules in a process like condensation of chromosome. So ಇಲ್ಲಿ cytoplasmic changes also occur in the prophase along with the changes that occur within the nucleus. So you'll see, I would like to introduce at this stage what is known as cytoskeleton anta. Cytoskeleton, that means every cell has got a cytoskeleton. Like human body has got a skeleton, every cell in our body has got a skeleton within the cytoplasm. That's why it is called as cytoskeleton, which is made up of three different kinds of molecules or the proteins. They are called as microfilaments, intermediate filaments, and microtubules. So, ನಮ್ಮ discussion based on the cell division, microtubules are very important for us for uh, you know understanding how exactly the chromosomes segregate to the opposite poles. So what you see here at the bottom, microtubule is a tubular structure, pretty long, whereas intermediate filaments are shorter. And of course, microfilaments are much shorter and also thinner. So when the cell is not dividing, these cytoskeletal structures occur and in the cytoplasm, and they are scattered in a particular fashion. I'll show you some of the fluorescent microscopic pictures, then you will understand what it is. So these are nothing but three different kinds of cytoskeletal structures. You can see on the top microtubule, which is coiled, and it's a kind of pipe. Okay, actin filament. They are microfilaments, and intermediate filaments are there that perform various other functions. Let us confine our discussion, our focus to microtubules, which is on the top. So there is a stage before the cell reaches metaphase from prophase, that is called as prometaphase. Prometaphase ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ the nuclear envelope will undergo disintegration so that the chromatin is suspended in the cytoplasm. Okay, so nuclear, it's a complex structure, as you know, bilayered, and many more structures are there. Nuclear pores are there. Nuclear pore complex is there. ಇದೆಲ್ಲ ಬಿಟ್ಬಿಡಿ. So just imagine how complex a cell is and how these complex structures are dismantled during the course of cell division. How they are reformed also is a big question, and a lot of research has been done on that, and so on and so forth. So it is totally disintegrated during mitotic division, enabling the suspension of this chromatin in the cytoplasm of the cell. So I'll skip this is nuclear lamina. When it is seen under electron microscopy, it will appear like a mesh. It appears like a mesh, and these structures are reformed once again once a nuclear pore, I'm sorry, nuclear envelope is made again. Okay. So the other component, what is important for us is the spindle fibers. Um, you know, the progression of mitosis depends on the formation of a spindle, which are present in the opposite poles. Now, this spindle is a transient structure. ಅಂದ್ರೆ ಇದು ಯಾವಾಗಲೂ ಇರೋದಿಲ್ಲ, but it is assembled during the course of cell division. ಯಾವಾಗಲೂ ಇರೋದಿಲ್ಲ ಅಂತಂದ್ರೆ existing microtubules are reorganized. I told you microtubules are a component of cytoskeleton. But during mitotic division, it so happens that these microtubules that are present in the cytoplasm scattered, they are reorganized in such a way that they appear as a spindle. They appear as a spindle, okay? Now, this spindle functions to separate sister chromatids to opposite poles. Anaphase movement of the chromosomes ಆಗಬೇಕು ಅಂತಂದ್ರೆ the microtubules should come into play. Okay, then only it is possible for the movement of the chromosomes or chromatids to the opposite poles, giving rise to two different nuclei, so that it divides to give rise to two cells from one cell. Okay. ಇನ್ನೊಂದು ಸೆಂಟ್ರೋಸೋಮ್ ಅಂತ ಇದೆ. ಸೆಂಟ್ರೋಸೋಮ್ is actually needed for nucleation of this spindle. So microtubules, if they want to get organized into spindle, it so happens that these components of the microtubule, they dissociate and then reform into specialized structures. ನಾನು ಇನ್ನೊಂದು ಫಿಗರ್ ತೋರಿಸ್ತೀನಿ. MTOC ಇದೆಲ್ಲ ಬೇಡ, ಅದನ್ನು ತೆಗೆಯೋಣ. Once. Yeah, see here. This is a cell which is in the interface where the microtubules are distributed all throughout. Okay. ಸೊ ಅದು ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ ಸ್ಪಿಂಡಲ್ ಆಗಿ ಪರಿವರ್ತನೆ ಆಗುತ್ತೆ. ಯು ಕ್ಯಾನ್ ಸಿ ದಟ್ this is the same cell where microtubules are reorganized to give the shape of a spindle. So this is a common picture that you have come across, that you have seen in your textbooks, where the formation of the spindle is very essential for cell division, especially dragging the chromosomes from the metaphase plate to anaphase to the poles, so that two cells are formed. Okay. So idu fluorescent microscopic pictures of the cells that are in resting stage and the same cells when they get into dividing stage, what changes can be observed. So you'll see here this green color, they are the microtubules. Fluorescent microscopy, with the help of fluorescent microscopy, these photographs have been made. Then here the microtubules are labeled with green color, and what you see in the center, that is blue color, is the nucleus. Okay. So at the bottom of this panel, you'll see that the asters are already developed, and you can see from each centriole which are present on the opposite poles, lot of microtubules are organized in the form of a spindle, so that one end of these microtubules are coming in contact with the chromosomes that are in the center. So can you see chromosomes? I am sure you can see the chromosomes in the center, and these microtubules are in the vicinity of the chromosomes. On one side, they are associated with centrioles, and on the other side, they are associated with chromosomes. So these two dense arrays, they are called asters, and the asters will also contain the microtubules, which are called spindle fibers, and those spindle fibers will attach to chromosomes. So metaphase ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ನೋಡೋಣ. So once again, I have put this particular figure to show what's the kind of organization of microtubules in the interface, prophase, and prometaphase, and metaphase. So I hope it is clear. At the bottom of these photographs, you'll see diagrammatic representations of what's going on as far as the spindle is concerned. You can see in the first one, the entire chromatin is within the nucleus, and in the second one, there is formation of asters, okay, and the chromatin within the nucleus is condensed actually, not to a great extent, but at least there is a little bit of condensation, okay. In the next phase, pro-metaphase, you'll see that there is disintegration of the nuclear envelope, and at the same time, some of these spindle fibers or the microtubules are getting associated with the chromosomes which have undergone condensation. So in metaphase, what happens, you must have seen in textbooks and also in your lab, that all the chromosomes of a dividing cell are arranged at the equatorial plate, and where they are associated with the spindle fibers. Okay, so that's how the chromosomes, they are aligned at the metaphase plate in the center of the dividing cell. There they get associated with the spindle fibers or microtubules, and then after they associate, you can see here the diagrammatic representation of a chromosome which has condensed, and the two parts of this chromosome is known as chromatids. This is one chromatid, this is another chromatid. So they are called sister chromatids. Sister chromatids ಅಂತಂದ್ರೆ the chromatids that belong to the same chromosome. Many a times, we also refer to as non-sister chromatids. Andre. Non-sister chromatids are the chromatids which belong to two different chromosomes or many number of chromosomes. So chromatids that are present in the same chromosome are called sister chromatids. So this particular chromosome, which is condensed, okay, is associated with cohesins. I told you they have the proteins that are present along with the chromosome, condensed chromosome, and cohesins. Cohesins actually they hold the chromatids together until they start moving apart. Okay, there are some forces which destabilize the cohesins, as a consequence of which they start segregating apart. Okay, and other structure which is very important as far as chromosome is concerned is the kinetochore. Kinetochore is a differentiated structure. Say in mitotic cell division or cell division dynamics, we often get confused with centromere, kinetochore, and centriole, etc. So you should be very proficient about it. Centromere is a condensed chromosome region which appears as a constricted region in the chromosome. That is centromere. Centromere, if you go to the ultrastructure of the centromere, although it is made up of cend-DNA ಅಂತ ಹೇಳ್ತೀವಿ, specific sequences of DNA are organized in such a way to give a heterochromatic region called centromere. Apart from centromere, kinetochore anta, that actually is a transient structure once again. Kinetochore is developed during cell division. So after cell division, the kinetochore disappears. So kinetochore ಏಕೆ ಅಂತಂದ್ರೆ, this is a differentiated structure to which the microtubules will bind. Okay, it's like an anchor. So it binds to the microtubules. You can see here, microtubule is bound to kinetochore. That's how they established the uh, um, relationship with the condensed chromosome. Okay. Hmm. So, so as soon as the microtubule binds to kinetochore, the chromosome moves towards centromere. Okay. It so happens that one particular chromosome is bound by the microtubules that are coming from the opposite regions. Okay, then only it becomes stabilized. Otherwise, it will be going towards one pole. So soon after, every chromosome is associated with microtubules that are coming from the opposite sides. It so happens that equal force will be operating on every chromosome, and that's how you'll see the alignment of chromosomes at the equatorial plate, which is nothing but the center of the dividing cell. Okay, so that's how we see in metaphase all chromosomes are arranged in one particular plane. Okay, so after that, it so happens that there will be division of the chromosome to give rise to sister chromatids, or the sister chromatids, they dissociate from each other because of this cohesin degradation of the cohesin. Okay, so you'll see anaphase is characterized by movement of the chromosomes to the opposite poles. So ಅಲ್ಲೇನಾಗುತ್ತೆ, these are different stages of pro-metaphase or pro-anaphase ಅಂತ ಹೇಳ್ಬೋದು. Until the equal forces are uh, established on the chromosomes will be moving in the cytoplasm freely. Now, once every chromosome is associated with the microtubules coming from the opposite sides, then it forms a metaphase plate. Okay, this is how this is a diagrammatic representation of
ಎಕ್ಸ್ಕ್ಯೂಸ್ ಮೀ,
the, yeah.
ಸಮರಿನ್ ಬೇಗಮ್ ಅಂತ ಹೇಳಿ ಒಂದು ಸ್ಟೂಡೆಂಟ್ ವಾಟ್ ಈಸ್ KMTS ಅಂತ ಕೇಳಿದ್ದಾರೆ ಸರ್.
KMT, ಈಗ MT ಅಂತಂದ್ರೆ ಮೈಕ್ರೋಟ್ಯೂಬ್ಯೂಲ್, KMT ಅಂದ್ರೆ ಕೈನೆಟೋಕೋರ್ ಮೈಕ್ರೋಟ್ಯೂಬ್ಯೂಲ್ ಅಂತ. ಅದಕ್ಕೆ ಬೇರೆ ಬೇರೆ ತರ ಮೈಕ್ರೋಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಇದೆ. ನಾನು ಆ ಸ್ಲೈಡ್ ಅದು ತೆಗೆದಾಕ್ಬಿಟ್ಟೆ, ಅಷ್ಟು ಡೀಟೇಲ್ ಬೇಕಿಲ್ಲ ಅಂತ. ಸೊ ಜಸ್ಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ರೈಟ್ ನೌ, KMT means kinetochore microtubule. That is a microtubule that is associated with the kinetochore of the chromosome. Okay.
ಓಕೆ, ಪ್ಲೀಸ್ ಕಂಟಿನ್ಯೂ.
Yeah, thank you. We'll not go into the details of these things because it is too much. And see, these are the things why I brought these diagrams and other details to you, is that there is so much to know, especially not only cell division, but in every aspect of what is going on within the cell. As much as you probe, or as deep as you probe, there is information, there is something to understand, which ultimately gives us a hand to master everything. So these are all the ways in which that you can contribute at the molecular level, at the cellular level, at the structural level, and so on and so forth. This is how research is being done, and this is how many small things, small results that are obtained are put together to understand the entire process. ಅದಕ್ಕೋಸ್ಕರ ನಾನು ಈ ಡೀಟೇಲ್ಸ್ ಎಲ್ಲ ಹೇಳಿದೆ, ಬಟ್ to explain all these details, it is not necessary for you right now. This particular interaction is only to activate your thoughts, activate your interest, activate your enthusiasm, so that you can think of going into different levels from this stage onwards, and plan your future. Okay. See, at the molecular level, see a diagrammatic representation how exactly the microtubules become shorter and shorter. See, if the chromatids have to move to the opposite poles, the fibers that are attached to these chromatids have to become shorter and shorter, so that the chromatids move to the opposite poles. Typically, what we call it as anaphase ಅಂತ. ಸೊ ಅದು ಅನಫೇಸ್ ನಲ್ಲಿ ಈ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಆಪೋಸಿಟ್ ಪೋಲ್ಸ್ ಮೂವ್ ಆಗಬೇಕು ಅಂತಂದ್ರೆ, these microtubules have to become shorter and shorter. Then only movement is possible. Okay. So along with the shortening of the kinetochore microtubules, you'll see various other kinds of molecules that are associated in this process of shrinking or shortening of the kinetochore microtubules. They are like kinesin 4, kinesin 7, kinesin 13. These are all, they are also called as motor molecules anta. Motor molecules ಏನು ಅಂತಂದ್ರೆ, these are the molecules that convert um, the chemical energy into physical movements. Okay, chemical energy into, I should say, movement of something which is associated with these molecules. Okay, chemical energy into mechanical energy. See, from chemical energy is nothing but ATP. ATP, the ATP is the input, that is energy, and that is converted into mechanical energy. And in that process, these motor molecules play a big role of movement of different kinds of organelles or different kinds of structures. NFS Gorna. I'll shorten this, it's already 5 o'clock. So NFS, you can see 3 different pictures. Now what you see on the top are the real pictures, which is taken under fluorescent microscope, where the microtubules are um, tagged with a green color, and the chromatin is tagged with a blue color. You see that this is how you will be able to see under the fluorescent microscope, and you can understand what else is going on here. And phase 1, and then telophase, and that telophase, that is the end of nuclear segregation. After that, there is another process which is called as cytokinesis, where the cytoplasm is divided, or division of the cytoplasm. Nuclear division is followed by division of the cytoplasm, and that is called as cytokinesis. These are independent processes. Andre, nuclear division need not be essentially followed by cytokinesis. Nuclear divisions may keep on occurring for a long time, giving rise to multicellular, multinuclear condition. Same cell will have multinuclear condition. At one point of time, I'll give you one example. At one point of time, there will be cytokinesis, so that as many number of nuclei are there, as many cells are formed. Okay, that's they are independent processes. But generally, when you say mitosis, nuclear division is followed by cytokinesis. But there are some examples where cytokinesis need not occur for a long time. Okay, and then at once it occurs. So here what you see is anaphase, movement of chromosomes. Telophase, you can see there is formation of nuclear membrane around the chromatin, and that's how nucleus comes into existence in the same cell, 2 nuclei, and this is followed by division of the cytoplasm, where you can see the formation of 2 cells from single cell. Okay, so for this process of cytokinesis, several other proteins are responsible. There will be formation of what is known as contractile ring. What you see in the red is formation of, until now it is not there. In anaphase it is not there, but in telophase, this contractile ring starts appearing. So this contractile ring formation actually enables the division of the cytoplasm, where you can see that there is uh, shortening and shortening of this contractile ring from a broad area. You can see that it becomes smaller and smaller and smaller and smaller. Ultimately, it will result in pinching off of 2 different cells from a single cell. By that time, actually, there will be formation of the nucleus. You can see the complete formation of the nucleus with nuclear membrane, etc., etc. And that's how cytokinesis results in the formation of 2 cells. So this is once again a diagrammatic picture. Let us not get into the details. So you'll see that kinetochore, right? KMT ಅಂತಂದ್ರೆ kinetochore microtubule ಇದು. where one end of the microtubule is associated with centriole, and the other end of the microtubule is anchored to the centromeric region or kinetochore region of the chromosome. Okay. So NFAs ನಲ್ಲಿ actually ಈಗ generally we can identify two NFAses. One is called as NFAs A and the other one is called as NFAs B. In NFAs A, there will be shortening of the KMTs, kinetochore microtubules, and in NFAs B, it so happens that there will be elongation of the dividing cell before cytokinesis occurs. So that is accompanied by elongation of the microtubules, another kind of microtubules called PMTs, polar microtubules. Okay, KMT shortening leads to anaphasia, where there will be movement of chromosomes to the opposite poles. But before cytokinesis occurs, it so happens that the distance between the nucleus increases. Although both nuclei are in the same cell, the distance between the nucleus increases, and that is a consequence of elongation of other kind of microtubule called polar microtubules or PMTs. Okay, and that signifies NFS B. After that NFS B, there will be formation of the contractile ring. Okay. Mm. See, this is how small experiments can be designed when you get into very specialized areas. You can see to understand what different kinds of proteins or what different kinds of environmental influences can be made on a particular process that is occurring. Here we take the example of a cell that is dividing. You can see that um, you know calcium ion concentration um, rises very rapidly in the dividing cell. You can see this is a small experiment. Specialized electron microscopic techniques, by means of specialized electron microscopic techniques, we have come to know that accumulation of calcium-rich vesicles occur at the spindle poles, at the polar regions. And this actually indicates whether or we um, it's an indication that these calcium ions, what are they doing in this particular region? So we suspect that possibly the calcium ion concentration is increasing in a dividing cell, which is not seen in the normal cell. Okay, and possibly this calcium ion concentration has got some role to play in the movement of chromosomes or in a dividing cell. Okay, so because when you compare to the regular cell, which is not under mitotic division, you'll see that there is a huge difference in the calcium ion concentration. So when the cell gets into division cycle, there is a rapid tenfold rise in the intracellular calcium ion concentration. So obviously, there is a jump in the calcium ion concentration in a dividing cell. So that actually, if you observe such kind of changes, you will start suspecting that this might be the reason, or this has got a role to play in a dividing cell. So Hagagi, further experiments are designed so that to understand why this calcium is rising, what is responsible for the rise in the calcium ion concentration. That's how we accumulate the data and then compile ಮಾಡಿದಾಗ ಏನೋ ಒಂದು ನಮಗೆ ಸಿಗುತ್ತೆ. And see, for example, in vitro cultured cells can be taken, okay, into which we can inject calcium ions. Micro injection of calcium ions is possible. Now, if we make such kind of changes deliberately, wantonly, experimental changes, cultured cells ತಗೊಂಡು ಅದಕ್ಕೆ ಕ್ಯಾಲ್ಸಿಯಂ ಅಯಾನ್ಸ್ ಮೈಕ್ರೋ ಇಂಜೆಕ್ಟ್ ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ ಅಂತ ನೋಡೋದು. Okay, so when that kind of experiment is done, we'll see that there is premature anaphase. Okay. So for this experimental cell, you should also have a control cell where you do not do anything in the normal cell. Okay. And keep observing both these setups, experimental cells and also normal cells to which no change is brought about. So this is a comparative aspect that everyone has to take in every kind of experiment. Control experiment ಅಂತಾನೆ ಮಾಡ್ತೀವಿ. Okay. So when there is micro injection of calcium ions, we have observed that these cells will get into premature anaphase before normal cells gets into anaphase. These cells would have already entered into anaphase. So this gives us a clue that these calcium ions are doing something, or they are responsible for initiation of anaphase. That's how inferences are made. Okay. So, okay, then shrinkage, etc., etc. I'll not get into the details of exactly uh, lengthening of the polar empties occur or PMTs occur. So that's all a consequence of what I told you is the activity of motor molecules, and that's how the chromosomes segregate apart. And cytokinesis, of course, you see the division of the cytoplasm. That's almost the end of my presentation. Different kinds of cells are formed. It may be radial symmetrical or spirally symmetrical or rotational, etc. I would like to draw your attention to the last one, last strip here. So you'll see that this is the embryo. This is the embryo um, with a single nucleus. Okay, this actually is occurring in many insects, especially in Drosophila. ಡ್ರೊಸಾಫಿಲಾದಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ, ಅದು ಆಫ್ಕೋರ್ಸ್ ಡಿಪ್ಲಾಯ್ಡ್ ನ್ಯೂಕ್ಲಿಯಸ್. This undergoes, this is after fertilization, etc., etc. Okay. When one cell divides mitotically, two, two to four, okay, and that keeps on dividing and dividing, or there will be nuclear divisions without cytoplasmic division. So you can see that this entire embryo is occupied with many nuclei. Okay, multinucleate condition, multinucleate. There is no division of the cytoplasm. So at one point of time, when all these nucleus, divided nucleus, occupies the peripheral region of this embryo, at once there will be cytoplasmic division, so that many cells are formed, which are now present at the periphery of the embryo. So this is how variations do occur. And this is the cleavage furrow, the dividing cell, where there is cleavage of the membrane to divide the cells into two. So this is a consequence of cleavage furrow formation is a consequence of formation of contractile ring, which is exactly in the center of the two center of the dividing cell. So this is actually diagrammatically represented here. You'll see microtubules, and in the center of the dividing cell there is formation of contractile ring. And if you look into the chemical composition of this contractile ring, it is made up of two proteins. One is myosin, the other one is axin. Okay, right? So um, these proteins are nothing but these are also found in our muscle cells. Okay, actin filament and myosin filament. Okay, so similar kind of contraction is necessary here. The mutual activation or mutual working of actin and myosin will enable the formation of contractile ring, and this contractile ring uh, contracts and contracts. You can see this reduction in the size of the contractile ring. Ultimately, it pinches off the dividing cell to give rise to two cells. So contraction is also seen in the muscle cells. You see, muscular contraction is a consequence of interaction between actin and myosin molecules. So I'll not go into the rest of the details. You can see here, this is a scanning electron microscopic picture of a cell that is dividing. Okay. Division is complete. Okay. And the same diagrammatically it is represented here. Yeah. Finally, I would like to conclude my talk with this statement. Fleming was the first person to give detailed description of mitotic events more than 100 years ago. I told you Fleming was the scientist who gave us the detailed description of mitotic cell division, prophase, metaphase, anaphase, and telophase. Yet, even after 100 years, we are still struggling to understand the relationship between different components of the mitotic spindle or the microtubule array that choreographs the movements of chromosome movements and formation of daughter cells. There are many, many details yet to be obtained. When we understand everything in total, then we may have a chance to control cancerous division of the cells. Okay. So with this, I would like to conclude. And if there are some questions, you may please address it to me.
ಸರ್ ತೇಜಸ್ವಿನಿ ಪಿಕೌಟ್ ಒಂದು ಕ್ವಶನ್ ಕೇಳಿದರೆ,
ಹೇಳಿ ಸರ್.
ಕ್ಯಾನ್ಸರ್ cells undergo rapid mitotic cell division. What is the reason for this, sir?
Cancer cells, okay. Now,
Yes.
cancer cells are the mad cells. Okay. ಅಲ್ಲೇನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, the regulation of mitotic division will be lost. Regulation of mitotic division will be lost. ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, I hope you can recall the cell cycle. I'll bring that back. Yeah. So ಇಲ್ಲಿ ಸೆಲ್ ಸೈಕಲ್ ಅಂತ ಏನಿದೆ ಕಾಣುತ್ತಮ್ಮ ಇದು?
Yes, sir, visible.
ಓಕೆ. ಸೊ ಇಲ್ಲಿ ಸೆಲ್ ಸೈಕಲ್ ನಲ್ಲಿ ಏನು ಅಂತಂದ್ರೆ, different phases are regulated actually. Different phases of the cell cycle are regulated by many, many factors, many, many proteins also. So G1S, G2 phase have to occur before the cell gets into division cycle or mitotic cycle. Okay. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, at the end of G1, at the end of S, at the end of G2, there are what are known as checkpoints. Checkpoints ಅಂತಂದ್ರೆ, those are the factors that will determine whether the cell has to progress in cell cycle or stop. ಅದಾಗ್ತಾ ಇರುತ್ತೆ ಟೈಮ್ ಅಂಡ್ ಅಗೇನ್. ಇಲ್ಲೇನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, ಕ್ಯಾನ್ಸರ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ, ದಿ ಚೆಕ್ ಪಾಯಿಂಟ್ಸ್ ಆರ್ they lose control over these checkpoints. Say for example, ಒಂದು ಚೆಕ್ ಪಾಯಿಂಟ್ ಏನು ಹೇಳ್ತೀನಿ ಅಂತಂದ್ರೆ, ಎಸ್ ಫೇಸ್ obviously is a phase where the DNA replicates, because say without DNA replication the cell cannot get into division cycle, because when the two nuclei has to be formed, earlier to the formation of two nuclei from a single nucleus, there should be duplication of the DNA. Without duplication of the DNA, it is not possible for a cell to distribute the genetic information to two different cells, which ultimately arise as a consequence of telophase. Okay. If the S phase has gone wrong, the checkpoint in the S phase will not allow the cell to get into G2 phase. So only when in the S phase, DNA replication is completed and the cell will sense that it is done already. So then it can allow the S phase to move into G2 phase. After G2 phase, that will get into mitotic cycle or dividing cycle, right? So M phase ಹೋಗುತ್ತೆ. So ಇಲ್ಲಿ at various stages G1, S and G2, there are checkpoints, and if these checkpoints malfunction, then the cell will become cancerous. ಇನ್ನೊಂದೇನು ಅಂತಂದ್ರೆ, there is genetic basis for cancer also, in the sense there are genes that control the division of the cells. ಅದು ಏನು ಅಂತಂತಂದ್ರೆ ಹೇಳ್ತೀನಿ, ಟ್ಯೂಮರ್ ಸಪ್ರೆಸರ್ genes ಅಂತ ಇದೆ. Tumor suppressor genes they are actually required for normal cell cycle. They are actually required for normal cell cycle. These tumor suppressor genes, if they mutate, if they undergo a mutation, if they become non-functional, then the suppression of the cell to become tumors is not prevented. Tumor suppressor genes, if they become mutated or if it malfunctions, then there is no check for a cell to divide. It keeps on dividing and dividing. ಅದೇ ತರ ಆಂಕೋಜೀನ್ಸ್ ಅಂತ ಇದೆ, ಪ್ರೋಟೋ ಆಂಕೋಜೀನ್ಸ್ ಅಂತ ಇದೆ. All these proto oncogenes and tumor suppressor genes are associated with the cell cycle. Any variation or any mutation or any factor that destabilizes these genes will cause the cell to get into perpetual division cycle. Although it's a very complex situation that you will be able to learn when you go higher and higher in your studies, especially ಈಗ molecular cell biology ಅಂತಾನೆ courses ಇದೆ. So where you will learn what are the molecules, what are responsible, how exactly molecular interactions will occur as far as cell functioning is concerned. ಅದು molecular cell biology ಅಂತ volumes of information and literature is there. A lot of research is done in the field of molecular cell biology to understand many molecular aspects and apply the same thing to for our benefit. It may be cancer treatment, or it may be some other disorder that is related to cell functioning, etc. So those are the implications.
So thank you, sir.
ಏನಾದ್ರು questions ಇದೆಯಾ? ಇಲ್ಲ
ಸರ್, ಬೇರೆ ಯಾವ್ದು ಕ್ವಶನ್ಸ್ ಇಲ್ಲ. ಸೊ
ಓಕೆ,
ಲೇಟರ್ ಆನ್ ಅವರು ಮಕ್ಕಳುಗಳನ್ನ ಕಳಿಸೋದು ಸಂದರ್ಭ ಸ್ಟೂಡೆಂಟ್ಸ್ ನಾನು ನಿಮಗೆ ಮೇಲ್ ಕಳಿಸ್ಕೊಡ್ತೀನಿ.
ನನಗೆ ಮೇಲ್ ಮಾಡಿ. ಅದೇ ನನಗೆ ಏನೋ ಒಂದು ಐಡಿಯಾ ಇರಲಿಲ್ಲ to what extent any subject has to be dealt ಅಂತ ಅಂದ್ಬಿಟ್ಟು. ಅಕಸ್ಮಾತ್ ನೀವೇನಾದ್ರೂ ಒಂದು ಪಾಸಿಬಿಲಿಟಿ ಇದ್ರೆ
ಸರ್, ಇಲ್ಲ ಇನ್ನೊಂದು ಕ್ಲಾಸ್ಗೆ ತಮಗೆ ನಾನು ಸರ್ ಜೊತೆ ಮಾತಾಡಿ ನಾನು ಕಾಲಾವಕಾಶ ಮಾಡಿಸ್ಕೊಡ್ತೀನಿ ಸರ್.
ಮಾಡ್ಬೋದು, ನಾನು ಇಮ್ಯುನಾಲಜಿ ಬೇಕಾದ್ರೆ ಮಾಡ್ತೀನಿ. ಎನಿ ಟಾಪಿಕ್ ವಿಚ್ ಇಸ್ ಇಂಟ್ರೆಸ್ಟೆಡ್ ಫಾರ್ ದಿ ಸ್ಟೂಡೆಂಟ್ಸ್.
ಆಕ್ಚುಲಿ ಮೋರ್ ದ್ಯಾನ್ ಮಕ್ಕಳುಗಳಿಗೆ ಸ್ವಲ್ಪ ಈ JEE, CET ಮತ್ತೆ ಈ ತರ ಒಂದು ಎಕ್ಸಾಮ್ ಗಳಿಗೆ ಸರ್ ಅವ್ರಿಗೆ ಅನುಕೂಲ ಆಗುವ ಒಂದು ರೀತಿ ಒಳಗೆ ಅವ್ರಿಗೆ ಮಾಡ್ಬೇಕು ಅಂತ ಡಿಸ್ಕಸ್ ಮಾಡ್ಕೊಂಡಿದೀವಿ ಸರ್. ಈ ಮಕ್ಕಳಿಗೆ CET ಗೆ ಮತ್ತೆ JEE ಬೇಕಾದಂತ ನೋಟ್ಸ್ ಗಳನ್ನ ಈಗ ನಾವು ತಯಾರು ಮಾಡಕ್ಕೆ ರೆಡಿ ಮಾಡಿದ್ವಿ ಸರ್. ನವೆಂಬರ್ ಒಳಗೆ ರೆಡಿ ಆಗುತ್ತೆ ನೋಟ್ಸ್. ಅದನ್ನ ಮಕ್ಕಳಿಗೆ ಡಿಸ್ಟ್ರಿಬ್ಯೂಟ್ ಮಾಡೋಕೆ ರೆಡಿ ಮಾಡಿದೀವಿ ಸರ್.
ಫೈನ್. ದಟ್ ಈಸ್ ಒನ್ ಆಕ್ಟಿವಿಟಿ ದಟ್ ಕ್ಯಾನ್ ಬಿ ಡನ್.
ಎಸ್ ಸರ್.
ಈ ತರ ಏನಾದ್ರು ಇದ್ರೆ ಐ ಆಮ್ ಫ್ರೀ ಟು ಶೇರ್ ಮೈ ನಾಲೆಡ್ಜ್.
ಖಂಡಿತ ಸರ್, ನಾನು ಇನ್ಫಾರ್ಮ್ ಮಾಡ್ತೀನಿ ಸರ್. ಸೊ ಎನಿವೇ ಐ ಹ್ಯಾವ್ ಟು ಗಿವ್ ಎ ವಂಡರ್ಫುಲ್ ಇನ್ಫಾರ್ಮೇಟಿವ್ ಡೆಲಿವರ್ ಟಾಕ್ ಆನ್ ಜೆನೆಟಿಕ್ಸ್. ಸೊ ಮಕ್ಕಳಿಗೆ ಬಾರಿ ಸರಳವಾಗಿ ಕನ್ನಡ ಮತ್ತು ಇಂಗ್ಲಿಷ್ ಎರಡರಲ್ಲೂ ಕೂಡ ಬಾರಿ ಸರಳವಾಗಿ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದಾರೆ ಸರ್. ಸೊ ಬಿಹಾಫ್ ಆಫ್ KSTA ಮತ್ತೆ ಮೈ ಸರ್, ಥ್ಯಾಂಕ್ ಯು ಸರ್.
Thank you very much. Thank you, guys. Wish you all the good, all students here. Thank you.
So thank you for all the participants, and thank you once again our CEO sir and our director Ravi Basaraj sir. So thank you sir. Thank you.
Welcome.